Okay. So thank you, Anpo. And actually, I realized when I listened to Roland's presentation, actually, my presentation should have been scheduled before his talk because I'm going to show the entire history of how we set the platform and how the recent progress happened. But anyway, it's my talk is not only covering tomato, but I also wanted to show some of the long-term work that we did for the past 50 years. If you recall the comment from Dr. Dennis Wong on the inaugural session, working on insect resistance is not so easy. And it's almost like looking for a needle in the haystack. And when I was studying as an undergraduate student in the university, the most striking examples that we used to get for host plant resistance always come from rice because of the extensive work carried out at IPRI especially for the brown plant hopper. They went on introducing the new genes after getting every new biotype getting evolved in the brown plant hopper. But what I want to highlight here, most of the successful examples for the resistant varieties to insects have come from the staple crops, the cereal crops, or the forage crops. And if you compare it with the vegetable crops, especially the commercial vegetable crops, it's just very little. So I was always asking, why is that? For example, if you look at one of the statistics, over a period of 75 years, there were hardly like 1100 publications on insect resistance. And it means 15 research articles in a year, maybe one in a month. So it's very meager. So I always ask why. So, okay. So as I said on the day one, we have a huge treasure box here. And unfortunately, we couldn't schedule a visit to the gene bank because it's a big team to get into the gene bank. And of course, we are always proud to be the largest owner of the vegetable gene bank among the public sector organizations. And it's amazing to have like over 65,000 accessions covering about 133 genera and about 330 different species. But if you look at some of our vast collections, go in Vigna, Solanum, soybeans, brassicas, capsicums, okra, etc. So most of our insect resistance work were dealing with these particular crops. And I just pulled out 3 examples for legumes because if you look at the diversity of insect pests on any particular crop, especially in the tropics and subtropics, it can range anywhere from 5 to 50. So which one we need to prioritize? Obviously, the one which need to be addressed as a high priority. In case of legumes, bean flies are the predominant pest problems. There are 3 species. And why they are the most important pest species? Most of the time, their infestation is limited to the first 30 days. Just when you get the cotyledon leaf, the infestation starts. And if you don't control them, you lose the crop completely. And in case of legumes, mostly it is grown under the marginal lands, under the rain-fed environment. And if the farmer, imagine if he or she loses the crop, finish. Because it's like you miss the season. You can't delay it for another, like a month, then you cannot go for another planting. So this is why we started focusing on the bean flies resistance in mung bean, because mung bean is covering millions of hectares in Asia, especially South and Southeast Asia. So as I said, the three different species of bean flies are quite important. And we had over 4,000 genotypes in mung bean. So we started screening them. The entire resistance screening and resistance breeding program covered a period of over 17 years. And as I said, we did the shuttle screening because we wanted to take advantage of the hotspots in the neighboring countries so we can expedite the process of identifying something meaningful, and then we can expedite the breeding pipeline. So then we identified the mechanisms, what contributes for the resistance, and then we passed the information to the breeding team. Then the breeding team starts using the resistance materials, and then we get the breeding populations and again screen them and the hotspots. So it goes hand in hand. And then we try to develop the bean fly resistant varieties. Then luckily in mung bean, we were able to develop a resistant variety. And if you look at its yield potential, it's almost 2 to 3 fold higher than the resistant parent. So even today, if you look at the average yield of mung bean, it's somewhere between 700 to 800 kilogram in a farmer managed production system per hectare. But this variety has a genetic potential of up to 1.75 tons per hectare, almost like double the yield. So since then, we started utilizing different sources of resistance to continue the program. And the next most important insect problem in mung bean is the storage pest. And why we wanted to focus on it, the farmers, if they are not able to manage this insect, they are forced to sell soon after the harvest. And the intermediaries and the buyers take advantage of this situation, and just they offer you the lowest price, lowest possible price. So if you want to increase the remuneration for the smallholder farmers, you need to build their capacity to store this crop at least for a few months, so their bargaining ability gets increased. So we wanted to focus this insect. There are two species in brookade, Calosobruchus chinensis, Calosobruchus maculatus. Both of them are quite predominant. And unfortunately, the farmers cannot notice the initial infestation, which is starting from the field at the harvest stage. If you keenly observe, there is a small egg, tiny egg attached to the surface. So when they don't dry the grinds, and if the moisture content is still like 8 to 10%, this is enough for the brocades. And then you put them in the tiny bags, keep it in the corner of the house. After 3 months, you see the mung bean powders. At least 2, 3 generations completed on it, completely crushing the grinds to powders. So we were looking for the sources of resistance, again, the same pathway, resistance mechanisms, and then passing the resistance sources for breeding. So here we used like more than 1,800 genotypes. But these genotypes come from different backgrounds. It's not only the Vigna radiata, which is the mung bean. We used another source from a wild mung bean, which is Vigna radiata sublobata. And it has excellent resistance, almost immune. But unfortunately, it has some undesirable traits like anti-nutrients for the humans. So unless we get rid of those anti-nutrient elements, these resistant sources are not highly useful. Then we also found a resistance in a related species, black gram, which is called Vigna mungo. It's not only Vigna mungo, and also there is a wild species, Vigna mungo sylvestris. And again, these sources are excellent sources, almost like highly resistant, or the sylvestris one is immune to the brocades. And then there is a subgenus of Vigna ceratotropes. Again, they have a couple of amazing resistant sources, but they are not crossable with the mung bean. So it's not just the finding the sources of resistance alone. We need to pass so many hurdles if you really want to be successful in terms of introgressing these resistance into the useful allied cultivar background. And yeah, for brookits, we have few sources of resistance and we were making progress. But until now, after 50 years, we have just a few sources. We have to rely on a few sources. And if the resistance breaks down, I don't know what to do. Okay, the next story is bean fly on the vegetable soybean. Again, the same issue because the vegetable soybean is a predominant crop, especially the East and Southeast Asian countries. And here also we worked for almost like 2 decades, worked with close to 7,000 genotypes. And again, shuttle screening happened. And we identified the resistant genotypes and involved them in the resistance breeding program. But one of the limitations is the smaller pot size and the seed size. So the horticultural trait is still not up to the expectation of the consumer. So still, this is an impediment to this particular resistance breeding program, I would say. Okay, let me move to a major story, the tomato, because this is probably the number one fruit in the world. I don't know how many of you consider tomato as a fruit, but botanically it's a fruit. So it's the number one fruit. And obviously, its cultivation is happening all over the world. And here in Asia and also in Africa, one of the major limitations is the tomato fruit borer, Helicobacter parmigira, which is highly polyphagous pest. But anyway, this is a major problem during the fruiting stage, the reproductive stage. So the farmers lose the income quite easily to this crop. So we wanted to focus on this particular pest because of the vast genotypes we have in our possession. So I would classify the breeding program, resistance breeding program, into 4 major groups. So we started with the Solanum hebrochetus, glabrum type. So in the initial screening, the 2 genotypes that we received from the North Carolina State University, they stood out extremely good. And they do possess the glandular trichomes, which produce the methyl ketones, especially the 2-tridecanone. But another disadvantage with these materials, yeah, they have high resistance, but they are negatively correlated with the fruit size. So if you want to increase the fruit size, which is preferred for the fresh market tomato types, and automatically the resistance level is going down. But anyway, we try to address this issue. We try to improve the resistance level and also increase the fruit size to at least to a moderate level. And our breeders were quite successful. And then at least we shared a couple of varieties with the National Agriculture Systems in Asia and Africa. The second group involved the Solanum pennellii, and Solanum pennellii is also having the glandular trichomes, and they produce the acyl sugars. And so we involved this particular accession LA716, which is an excellent source of resistance for whitefly, as well as a couple of other foliage feeders. And then we developed the backcrossing materials. But again, there are some undesirable traits like genes conditioning the resistance, the high acyl sugar. They are also causing the low pollen fertility and lead to small fruit size and other undesirable horticultural traits. So the resistance breeding involving the Solanum pennellii was also paused for a while. Then in Solanum hebrochetus, another group, Typicum, and we had another excellent source of resistance, LA1777, and it's highly resistant to the tomato fruit borer. So we started working with the Cornell University, and Cornell University developed the near isogenic lines, and we got the near isogenic lines screened here in the tropical environment. But unfortunately, they don't provide a quite good control of this pest here in the tropical condition. So again, we passed this one. So the last attempt involved the Cerasiforme type. And then we got two good sources of resistance, which are known to be resistant to the beet armyworm in USA. So we tried it here against Helicoverpa. It was extremely resistant, and they produced the higher concentration of glycoalkaloids. So because of the previous experience based on the foliar-based chemicals, which are not leading to a desirable fruit size, we did not want to invest our time using this material in the breeding program. So it's a kind of like a sad end for all these efforts over a period of like 2 decades. So there was a pause of 10 years. Then back in 2012, there was a publication from Wageningen. So when I saw this paper, because all the work I mentioned was carried out by my predecessor, Dr. Talekar. Some of you might know him. So when I took over the program, so when I saw this publication, I thought, okay, why don't we revisit our gene bank for the possession of the species mentioned in this publication, which is Solanum galapagense, which Roland was highlighting in his presentation. So we identified a few accessions originated from Ecuador. They were present. So immediately we were screening these accessions in a no choice condition for 2 years in 2013 and 2014. And 2 of the accessions were found to be excellently resisting the whitefly development, feeding and development on them. So this is the result from 2013 and this is from 2014. So subsequently, we got Muhammad Raka, who was working with us for a couple of years. So we started working together, and besides Solanum galapagense, we also involved some of the accessions from Solanum chismani and Solanum pimpinellifolium, and we were focusing on multiple insect pests, not only whitefly. We focused on thrips, we focused on the Lirio leaf miner, and later with the arrival of Tuta absoluta, Thorimai absoluta, we also used them. And also we tried against the 2-spotted spider mite, or red spider mite, Tetronychus species. And we did extensive work here in Taiwan and also in Tanzania and Kenya and East Africa. And because of the time, I didn't go through all the results, but if you are interested, you can look at these publications. We had like 3-4 papers based on all this work. But what I want to highlight here is the basis of resistance. So this is an example from our recent work on Tuta absoluta involving some of the accessions like Galapagense, Chismene, Hebrachaetus. If you look at the cultivated tomato and these wild species, they reduced the number of eggs laid. So they have like the oviposition repellency. They have strong anti-feedance effect against the young caterpillars. So they resist the Tuta absoluta like anything. And then when we studied what contributed to the resistance, they are glandular trichomes. Probably you might be aware the Solanaceae, especially the tomato, has a range of different types of both glandular and the non-glandular type of trichomes. So what contributes mainly for the resistance are the 2 types of trichomes, type 4 and type 6. So type 4, if you look at the Galapagense and Chismeniae, it's primarily having the type 4 glandular trichomes. And then the Hebrachaetus, they have type 6. Especially in type 6, there are like two types. One is the single lobed, the other one is the four lobed. So they possess the high amount of the type 6 trichomes. So these glandular trichomes and the chemicals produced like acyl sugars in those glandular trichomes are primarily contributing for the resistance. So then you hear the story from Roland, which was carried out based on these works to what we are today. So then quickly, I want to go through few other projects that we carried out in the last 20 years. Thrips resistant onions. So thrips, which is a cosmopolitan problem on onions. And for the last 20 years, they are also creating a huge havoc because of their transmission of the iris yellow spot virus disease. So in Africa, onion is a very important crop, and they don't have too many varieties, and even those varieties are not resistant to the thrips. So we worked with the ISIPE, International Center of Insect Physiology and Ecology, headquartered in Nairobi, Kenya. Over 3 years, we screened about 160 onion germplasm materials against Thrips tabaci. And then we found two varieties or two accessions. They have excellent resistance besides a very good horticultural trait, which is a very nice bulb size. So even they were producing higher yield, a bigger bulb than the very popular variety, which is commonly grown in East Africa, which is the Bombay Red onion variety. So we introduced these two lines in Africa. And of course, one of the major factors contributing for resistance is the higher amount of epicuticular wax. So one thing I want to highlight here, if you look at this picture, most of the existing literature so far, they showed the amount of epicuticular wax is positively correlated to thrips. If you have more amount of wax, you get high thrips infestation. But in our case, it was the reverse. So this is the scanning electron microscopic picture of the surface, leaf surface. Yeah, leaf surface showing the wax particles, the different wax types, and also the wax arrangement. So if you look at the resistant genotypes, the wax particles are very densely packed. There are different types of the wax particles. They are very densely packed and they are highly resisting the thrips infestation. So this is just a contradictory result that we got based on our studies. And two last examples. One is the aphid resistant okras. Again, okra is an important crop in some of the Asian countries and also most countries in Africa. So we screened the okra germplasm involving two major species, the Abelmascus esculentus and the West African okra type, which is Abelmascus calei. And then we were able to identify a few resistant genotypes which fit very well across the different agroclimatic conditions in Central Africa condition, because the major focus was Cameroon in Central Africa. And then we introduced these varieties into Africa. Then the last example is coming from the amaranth, because in amaranth, one of the major pest problem is the amaranth leaf weber, which is a defoliator. But besides the leaf weber, we also have a wide range of weevils, especially if you grow the amaranth for the multiple cutting, or if you grow the amaranth for the grind type, which stays in the field for several months, you get the weevil infestation. So we screened the varieties for the two different insects, and we identified a few, very few resistant genotypes, and we introduced them into places like Tanzania. So as Roland highlighted, insect resistance is one of the traits. And then we always try to embed them with the resistance to other biotic and abiotic stresses. So we want to provide the holistic solution to the smallholder farmers in Asia or Africa or in other parts of the world. So what kind of impact these varieties can create? So based on a few impact assessment studies, I want to show you just 2 slides. This is the impact that we created by introducing the improved mung bean varieties in different parts of Asia. And also in tomato or pepper, if you look at the varieties available or the acreage covered, at least 15% of the varieties are from world veg. And the last slide is the impact coming from Africa. You can see the different crops like tomatoes or African eggplant. In case of African eggplant, almost like 100% of the seeds are from world veg. The varieties are from world veg. And in case of amaranth, in fact, this is even much higher because this figure has come from the socioeconomic survey. But recently, we also conducted like the molecular phenotyping, because you randomly go around, collect the samples from the varieties grown among the farmers, and you phenotype and genotype them, and then you can find the correlation to the varieties generated from world veg. So even it's like almost over 60% now. So to conclude, what we want to see is the smiling faces among the smallholder farmers around the world through the consistent work that we carried out using the treasure box sitting here, because the seeds are the easiest technology to adopt by anybody than any other technology. So this is where the insect resistance work is quite important. That's why we are all here today. Thank you.